Okay, so since it's 3 o'clock, I guess we'll get started. So this lecture is a continuation of my last lecture 14, where we went over some of the basics of alignment. And today we're gonna try aligning some data ourselves. Okay, and then we're gonna explore visualization options. Last class, we downloaded the different references using this slide here. And today we're going to work with some chip sequencing data. And then next class, Igor is going to go over RNA sequencing data. So the chip sequencing data that we're going to be working with is from ENCODE. Here are some links telling you about the data. If you're interested, I've got a Geo link, the SRA link. And I want to give a disclaimer, we aren't analyzing the data as you should with a proper chip sequencing experiment. I'm not going over controls or peak finding or looking at comparing the control to your condition. This is just aligning something to the genome and then visualizing it. And I think that should take about the hour that we have. Okay. So the first thing we're going to do is we're going to download our data and run FastQC on the data. So we're going to start from the beginning. And some of this you went over with Lisa, so we're going to go over this fairly quickly. And if you want me to slow down, just shoot me a text in the chat and we'll slow down. So let's move to Big Purple. Okay, I'm on Big Purple, and now I'm going to move to our class directory, which is GPFS. Okay, so we're in our class directory. We're going to move into alignments. And then within alignments, I have 3 different directories here. I have a data directory, I have a references directory, and I have a scripts directory. And I forgot to give you all permission to the directories. Okay, so you should have permissions to copy everything. You don't have permissions to edit the things in this folder. And we're going to move it into our scripts directory. And this is where I've put all of the pre-written scripts that we're going to go over today. And since we can't, or since you can't write within this directory, we're going to copy these files as we go into the directory that you're going to be using for this tutorial. So I've given you an example of how this will work here. So you're going to use the CP to copy the file. The first input to the command is the path to the file you want to move. So we're going to move the download examples.sh file to whatever path, your home path. If you created a new directory for this class, move the file to that directory now. The way I've formatted the slides is I have the copy command. I have an example, just broad strokes of how you would use the functions, the files that I've created. And then I've given you exactly how I use them, but it's not going to work for you if you copy and paste this because you don't have access to these paths. So you're going to have to change it yourself. And then here, I know some people didn't have access to Big Purple. You might be on prints or something. So I just copied and pasted the code onto the slides if you were curious. And then there are some links that if you follow will give you some extra information. So this is a link that'll take you to some explanations of FastQC, which we'll see in a second. Okay, so we're downloading our example. So let's look at what's in download example. Okay. So last class we learned about variables, and I'm using variables heavily in these files that I've created, because it's just going to be too much if you have to delete my paths and input your paths. So here we have our first input, the out path, and our second input, the SRR name. The out path is where you want the data to go, and the SRR name is the SRR file here. Here we're loading the required modules, so we're loading SRA Toolkit and FastQC. This is really, I copied the code from Lisa's module and just added a few lines. And then here I'm checking to see if the path you supplied doesn't exist. So if it doesn't exist, it's going to make that path. And if it does exist, you're just going to use that path. And then you're going to download the data like you did with Lisa, where your output now is your out path that you specified with your first variable, and then the actual SRR file name is the variable that you specified as your second variable here. And then in the same file, in the same workflow, we're going to take that copied, that downloaded data, and we're going to run FastQC on them. So we're going to create a new path that's called QC within our out path. And then we're going to run FastQC here. The out path is now this new path that's called QC. And your input to FastQC is the SRR file that you downloaded. So if we go back to the data and go to raw for now, this is what the output is going to be here. So we have these two SRR files, so they end in 222 or 223. So you have 222 and 223. And then if we go into QC, we have our FastQC for 222 and our FastQC for 223. And the way you would run this is to sbatch your command like so. So nothing is being done on the head node. And I've created sbatch files for everything, even for some of the jobs that are a little smaller, because I don't know if we would all get srun jobs if we were doing this interactively. Okay, so I'm going to give you a second to run that. I'm not sure how long it'll take everyone. But I'm going to assume that you ran sbatch, download example, the path where you want it to go, and this SRR file. So we're using these two here, SRR 10838222, and the one that ends in 223. Okay. Now, since you can't read the HTML files so easily here, we're gonna copy the data to our local computers and view it from there. So if you remember to copy the data from Big Purple onto your local computer, you're gonna go to your local computer. So I'm gonna create a new tab by doing Command T. So now I'm on my local computer and I haven't logged on to Big Purple or anything. So I'm going to use SCP my KID, AY1392, so I'll type it with you, 92. And then the path to the data. So you guys can copy things, I'm pretty sure, to your home. So copy the fastqc file. There's no such path or directory. Let's see. Okay. Courses. BI. Okay. And let's just copy and paste because that's way easier. And then I created a folder for myself. Within my local computer, and I downloaded it. And then now we can go check to see what that looks like. So I downloaded all of the FastQCs that we will look at, but this is the one we just downloaded. We go through the base statistics, they look fine. And we got checkpoints, check marks everywhere, except for here. We have an overrepresented sequence. It says it's a TrueSeq adapter index 4. So that indicates that there are still adapters on these reads and we need to trim them off. And if we look at the other one, 223, we see the same thing here. So we have to go trim off those adapter sequences. Okay, but before we trim the adapter sequences, we're going to concatenate these two FASTQ files because they come from the same sample into one FASTQ file. So the way we're going to do that is we're going to use this file I created called concatenate FASTQ, and that is really just using the function concatenate. So if we go into scripts, you have to, again, copy this concatenate underscore fastq.sh into your own directory to be able to use it. So if we look at concatenate.fastq, has the normal headers that we have for an SBatch job, and it takes in 3 arguments. It takes in the first SRR file, or the first FASTQ file, it should be, FASTQ1, FASTQ2, and then the name to name the output FASTQ. So if we go through, we're using the function cat to concatenate these 2 files. It'll concatenate FASTQ1 with FASTQ2, and this pipe, this just means pushing the output into this file here. This file has the third variable, so starts with your third variable, whatever you put in third, and ends with .fastqz. So if we look at my example, what I did was sbatch concatenate fastqc fastq.sh, then the path to the first FASTQ file, the path to the second FASTQ file, and then the path up to the name of the output concatenated function file. Okay. So if you do that, we're still in raw. We have our file here that is concatenated. And if we look at the size of the files, you can see that the two original Fescue files are roughly half the size of the resulting Fescue file. So we've taken two Fescue files, we put them into one, and now we're going to use that one concatenated Fescue file moving forward. And I'll give everyone a few minutes. Is everyone able to follow this and run this as I go? Or am I a few steps ahead of everyone? Okay. So I'm going to move on. And we're going to remove adapter sequences using trim galore. So we saw in Ucsc that there was a lot of sequences from adapter sequences from TrueSeq. And I looked online, I searched around and saw that this tool trim galore will get rid of Illumina adapters automatically if you specify the flag Illumina. So that's what we're going to do. Francesco asks, yeah, they're the same. They're from the same cell. Like they are, yeah, they're from the same experiment. So we're going to trim our adapter sequences now. And we're going to use the function, the file that I created, trim.sh. So if we go into trim.sh, you have to copy this again to your own directory. So here is how you would copy it. And then our trim.sh function takes three inputs. It takes the path to your data, the name of the Fescue file, basically, and the output path, so where you want to put it. We're going to load in the required modules. We have trim galore, Python, and Fescue. And here again, I did another check to see if our output path exists, or sorry, this exclamation point means does not. So we're checking if this output path here, if this directory does not exist. Then, so this is an if-then statement. We went over in R, but this is the bash version of that kind of structure. So let's walk through this. So we're saying if the thing within these brackets is true, then do these two lines, and then you basically finish and then keep going with the script. If it's not true, then you don't even go within here. So here within the brackets, we're saying if this output path, that's a directory, is not a directory, so this exclamation point means not, then you echo this path. Echo prints stuff out. So maybe you did this with Angelina. But if you echo test, you're going to print out test. So we're going to echo the path does not exist, creating this path. And then we're going to create that path. So now the path does exist. Now we're going to use trim galore. And we're going to take things that are good quality, and we're going to trim the Illumina reads off. And the way that we trim the Illumina reads off is trim galore looks for this sequence and then trims basically those sequences off. So trim galore also allows you to specify your output. So you're going to output a gzipped file, and then we're also going to run fastqc on this resulting trimmed file. And then we have our input Fescue, which is at our data path, and the file name for the Fescue file. So when I did it, the file name was srx7 or whatever the concatenated file name was. And then your output, so that's your -0-O for output. And then you specify your output path. So here in practice, what you would do is you would run sbatch trim.sh, your path to your data, which for me is the alignment data raw. So if we okay, I just took a lot of shortcuts there, but that's just so that I can get there faster. Okay, so this is where my data is. I'm giving it this name here. And then I'm giving it the path, the output path. So I said I wanted another directory called trim within the data file here, trimmed. And that's what's going to happen when you run this line. So I'm going to give everyone 2 to 3 minutes to run that. Also, if you're behind on the actual steps, you can catch up by copying the actual data because I've pre-ran everything. So every step is already run. So if you want to run it as we go, what you could do within alignment is you can copy all of the stuff within the data directory, or all of this stuff only within the raw directory or the trimmed directory to quickly catch up. So how you copy a directory is you do cp, but you need to specify that you want to do a recursive copy. Excuse me. You're going to give it the path to the directory. So right now it's just raw because I'm within the data directory. Then I need to give it a path to copy. So I'll copy it to my home directory, to my bi20 directory. It'll take a second. If we move to the bi20 directory, now you can see I guess I copied everything. I'm not sure if I copied that before, but here we have raw here. Oh, I know why that happened, because I created a symlink, but never mind that. I'll just let that be for now and clean it up later. Okay. So once we've trimmed our adapter sequences, now let's look at the resulting Fescue after we've trimmed the adapter sequences. So we have to copy the Fescue.html file onto our home computers. So to do that, let's go back to our home. To log out, I'm sure you know how to log out. I did Control-AD to log out. Now I'm on my home computer. And now I'm going to copy the Fescue file after trimming to my home directory. And I'm going to do that using SCP again. Okay. So we're here in the trimmed directory now. And my file is called trimmedfescue.html. Oops, I forgot to specify where I want it to go, so it didn't work. Oops, and I spelled Big Purple wrong. Big Purple. Okay, so I copied that to my home directory. Sorry, my local. And here, let's check it out. We no longer have overrepresented sequences like before, but we have some weird per base sequence content, and that's the only really red mark that we have. Other than that, we're okay. And this per base sequence content is an artifact of the trimming that we just did. So there's no real huge alarm bells there. Okay. So back to Big Purple. We have downloaded the data. We've done QC on the data. We've trimmed the adapters off of our reads. And now, we're going to use Bowtie to align our reads to the genome. So last time, we all downloaded the human genome reference from Ucsc or Ensemble. And we're not going to do this, and you don't have to do this because thanks to Sana, we learned about the iGenome module. So we're going to use the iGenome module, but I'm just going to show you how you might create an index using Bowtie in case you aren't on Big Purple or in case you're on your local computer and you need to create an index on your own. So let's go to scripts. And I created a script called Bowtie build index. So let's go into Bowtie build index. Bowtie build index. Okay. Here we have our header sbatch again. Our first input is the path to the genome FASTA file that you downloaded. And then the second input is the output path to where you want to save the Bowtie indexes. you're going to load in a bow tie, and then you're going to check whether the supplied path, out path, doesn't exist again. And if it doesn't exist, you're going to create it. If it does exist, you'll just write to that path. You're going to move into the path using CD, and then you're going to create the bow tie to index using the genome path. And then here, this is just the prefix for all the files that will be created. So I ran the bow tie path, this file here, and if you were me and you ran this, this is what your output would be. But let's go over how you might run this file on your own. So first, you sbatch. You have your file name. You have your first input is the path to the genome, the full path. The second input is the full path to where you want it to output. So for me, I said sbatch bowtie build index.sh. My input file was here. This is where I had downloaded the HD38 genome FA. So it's within the class directory, within alignment, within references. And then I wanted it to output a new directory. So it creates this directory for me within references called bowtie2. And this is the directory that we're talking about here. If we go within bowtie2, this is the bowtie2 indexes for the genome. However, this will take too long, and we don't need everyone to create their own references, so we're going to use iGenome. So let's check out iGenome on Big Purple. So if you type in module show iGenome, you're going to get this output on the terminal. And what this says, first, it'll tell you what this is. So these are the Illumina genome references, the genome sequence indices, and this is where it's found. So let's go here. The references are found here. So we're going to go here into iGenomes. We can see that there are many different species that are hosted or that are maintained. Let's go into Homo sapiens because that's what we're working with. Let's go into UCSC just because that's what we started with last week. Let's go into HG38. Let's go into sequence because we care about the DNA sequence right now. And we have these different options. We're going into the bowtie2 index. And here is the index for everyone to use. So we're going to use this path later on. So copy this path and save it because we're going to use it when we run our alignment. And there are different things you can do, like add the genomes to the path, but instead of going into different path variables, we're just going to use the straight direct path to exactly what we need. Okay, so we are going to now align the reads to the genome using bowtie2. So let's move back. Okay, so we are in GPFS data. Okay. Okay, so we're going to go into scripts. And let's take a look now at the bowtie align file. Okay, so here we have our standard S batch. We have only one input now, just the path to the Fescue file that we are going to align to. Oh, and I guess you didn't have to copy the path because I copied it and just pasted it here. So I didn't make the bowtie2 reference a variable. If you want to make it a variable, you just say bowtie2 equals $2. You get rid of this line and you add it as a second variable. Here we're loading in the bowtie2 module. I'm setting bowtie2 ref to the path that we just got to the bowtie2 index. And now we're going to align the reads from the Fescue file. So this Fescue file that we're using is our most recent Fescue file. So the trimmed, the concatenated and trimmed Fescue file. So we're going to call bowtie. We're using a local alignment. Let's talk about the easier things. So we're using the bowtie2 reference, that's this X, and this U is your input Fescue. This S is your output SAM, so this is the name of the SAM file you want to output, and then also we're going to output some of the things that are written out as the program progresses into a file as well. So let's look at the specifications of bowtie and look through. Maybe I didn't copy them onto the slides. Okay, so if we go to the bowtie2 manual, there are a lot of different options. And we can go through and see what bowtie does and what bowtie doesn't do. You should read this before you use bowtie. And then also, let's look at the bowtie2 aligner. Okay. Okay, here, this is what I wanted. Okay, so I wanted to show you guys how to look at the usages of things and how to figure out what people are doing and how what these flags mean. So each one of these flags tells the program some parameter. Here, let's go through. We have a -x. Our -x is here. And this is the base name of the index for the reference genome here. Here, okay. And I think I have to change this file because I didn't specify it all the way to the base name. So okay, so let's all change this portion of the file. So I gave the path to the reference genome, but it says it wants the base name of the index of the reference genome. So if you remember, the base name was genome. So we need to add that /genome part, or else it won't work. Okay, so then Control X, Y, and we have changed the file. So let's look. Okay. And then you can go through all of these flags and see what they mean. I'm not going to go through them right now because these are very ChIP-seq specific, but this is what people use when they do ChIP-seq. Okay, so then back to the lecture for alignment. Okay, so to align and use that script, this is wrong. I have an equal sign here. Don't use that part of the path. You're going to do sbatch, the file bow tie align, and then the path to the Fescue. And then for me, it was sbatch bow tie align, and then the path to the Fescue there. So the output of that is in data. Okay, so if you remember, I said that I wanted the output SAM file to say unfiltered underscore sam.sam. So that is this file. So this is the first file that results. So we have now aligned the reads to the human genome, and we have a SAM file. So the SAM file is the not compressed version, and so we're gonna want to change the SAM file to a BAM file, which is easier to work with. But before we do that, we want to be able to filter our SAM file to only keep the alignments that we want. And we're going to use SAMtools view to filter. And so I have the link to SAMtools here, but let's see. Okay, SAMtools is great. SAMtools helps you manipulate SAM and BAM files. These are all of the different types of things you can do with SAMtools. And then there are commands and I think different command options and how to use each of these on their own. Let's see if we click on one. Yeah, if you click on one of the options, one of the functions, it'll tell you the description and then the options for that specific function. Okay, so let's go into SAMtools filter, set SAMtools view. I'm not in the right directory. Move to scripts. Okay, so we're going to look into SAMtools view. And here, again, our header. If you noticed before, for the alignment and the index creation, the time I made to be 10 hours or something, which is more than was needed, and more memory. The first input to this SAMtools view function, this file, is your input SAM, your output SAM, your output BAM, sorry. You're going to module load SAMtools, and then you just run SAMtools view -b, which means you want your output to be in BAM format, -f, which is similar to the exclamation point. It's everything that's not in this bit flag 4. And then the output, and you have your input SAM. So your output BAM and your input SAM. And this 4 comes from the flags that the alignment flags that we talked about last class. Okay, so if I were to use this, again, you have to copy this to your own directory. Sbatch, SAMtools view, and then the path to the data as it is, and then the path to the output file that you want to create here. And I want to mention that I made all of these things sbatch files just so that we're not using the login node when we're running these examples. But if you're stuck if you need to debug things, the best thing to do is create an srun and then do this interactively. So you're going to module load and then play around with these things until it works for you, and do it interactively. Okay, so, oops. If you go back to the data, this is exactly what I wrote to get the output. So I said the input data is our unfiltered SAM, this one here. And then I wanted to create an output called output BAM here. So this is the one that's output. And we filtered for only reads that mapped to the genome, and that's our flag, or what we took, reads that map to the genome here. I put this queue here, this flag queue, in case you want to filter for alignment quality. So if you want to use the map queue, this is where you have to know your tool and the map queue values for the tool. So I think I have a link somewhere here. Someone's blog post that was really helpful in determining bow tie to map queue scores, which are different from the BWA ones and are different from the STAR ones. So if you want to filter based on alignment quality, then you should read his post and decide which ones you want to keep, which map queue values you want to keep, and then use the -q flag. Okay. Now we have aligned our reads. We have a SAM file. We filtered our SAM file, got a BAM file. Now what we have to do is we have to sort and index our BAM file. Okay. So I created a sorting and index file here. And I have a typo up there that's not good. Okay. Oh no, I don't have a typo. Okay, whoops, sorry, false alarm. Okay. I thought that was a different line, but it's just from this line up here, so that's okay. Okay, so we have an input BAM, we have an output BAM. We're going to module load our SAM tools, and we're going to use SAM tools sort. And SAM tools sort takes an output file and an input file. So we're going to give it our most recent BAM, which was output.bam, I think. And we're going to create a new BAM from the sorting, and then we're going to index the BAM file. Sorting and indexing a BAM file is really important because it helps, since BAM files are huge and you have a lot of reads, it really helps to sort and index them so that programs can easily query regions of the genome. And a lot of times, if you haven't sorted and indexed your BAM file and you go to the steps, the next steps, you won't be able to because programs expect a sorted and indexed file. Okay, so you're going to copy this to your own directory. To use the file that I created, you're going to sbatch samtools sort index, the path to the data. So in my case, it was output BAM. Let's look. We have output BAM here. And then we created a sorted BAM. Yeah, it's sorted by genome location. It sorts by genome location. And you create a sorted BAM here because I said that I wanted my output to be called sorted BAM. And then it creates an index for this sorted BAM here. So this .bai indicates that this is a BAM index. Okay. So we've done our alignment, SAM file, we filtered our SAM file, we've got a BAM file, we've sorted our BAM file, and we've indexed our BAM file. This is great. These are the main steps you have to do. And then the next is to visualize your alignments using IGV or UCSC, Genome Browser or WashU. So in the next few minutes, I'm going to show you how to change your BAM file into other file formats. And also, I also had a slide on peeking into our BAM file using SAMTools stats. Okay, so SAMTools stats will tell us more about the alignment. So far, we've aligned our files, but we don't really have we haven't done like a fast QC type thing to see an overview of how our alignment went. So let's look at this file I created called SAMTools stats. Okay. So the input to this file is just the input BAM. We're using SAMTools again, and this is SAMTools stats, takes an input BAM, and then we're using this caret thing that just means whatever the output is from this line, this code, we're pushing it into this text here. So let's see. Where I think it's this one here. Here. So we have our output from SAMTools stats. So it says this file was produced by SAMTools stats. And you can plot the data from in this file using BAM stats, plot BAM stats. And this is just a brief overview of what our reads look like. So see, we can see reads where the QC failed. We don't have very many. Duplicated reads. We don't have paired reads. So look, we don't have any paired reads. This is how many mapped, and this is how many total sequences we have. This makes sense because it makes sense that our total sequences and our map sequences are the same because we filtered for only mapped sequences when we converted from our SAM file to our BAM file when we did dash capital F4. Okay, so that's one way you can look at your alignment stats. Okay, a lot of times you might want to use files like bigwigs or bed graphs instead of BAM files. And bigwigs and bed graphs are great because they help you visualize continuous data. So let's look at this code that I wrote to convert BAMs to bigwigs using another set of functions from a program called Deep Tools. So let's okay, so this is the function that we're going to be using, BAM coverage from Deep Tools. On Big Purple, you can just module load Deep Tools. Deep Tools has a lot of other tools like correcting for GC bias. You can do plotting correlation, plotting the coverage, comparing BAMs, what have you. So Deep Tools is really helpful it's another way that you can manipulate BAM files and work with BAM files. Okay, so I used this code. This is saying BAM, we're gonna calculate the BAM coverage. Our input is a BAM here, the input BAM from your first variable, and then your output is gonna be that input BAM dot BW, and BW means a bigwig file. I made the bin size equals 50. You can change the bin size. The smaller bin size would be more granular. And then I made it verbose, which means it'll print out what it's doing as it goes so that I could keep track of what was going on. And then I also took this code from the BAM coverage example because it has some normalization steps. You can normalize to the effective genome size. And here I took the genome size. Here's the link, I copied it for a human. For the normalization here, we ignore chromosome X. And this is what people use often for chip sequencing. So bigwig, these different file formats are really similar, and sometimes I myself can get confused about why I need one file type over the other. But here, if we look, this link here tells you exactly which each one of these file types are, and you can read each blurb and it'll tell you more in depth about why you need each of the different file types. For instance, like a bigwig file, they say, is best for displaying continuous value plot data, such as depth of reads. Okay, so we have converted our BAMs to bigwigs here, and we have our sequence depth normalized bigwig there. So we are done with our alignment, and then the next thing to do is to visualize, but I took a little bit too much time. So I'm probably just going to end here, but give a brief overview of the different things you can use to visualize. So you can use the Integrative Genome browser. Let's see here. Here is the output sorted BAM here that I just uploaded. You can upload data from your computer. It's really easy. You have different tracks that you can overlay and see, if your reads overlap with genes. You can use the Ucsc Genome browser. And if you want to see cool things people are doing with Ucsc Genome browser, there's a page with public tracks. So you can see, here's someone from NYU, maybe you know them. He's looking or she's looking at this region of the genome. But if you see the differences between UCSC and IGV is that you can share the things that you're looking at with people, whereas IGV you can't really. And then Igor also pointed me in the direction of WashU Genome browser to show you guys. Which is good for, there's a new one. Maybe things like Hi-C that you can plot here. And then lastly, if you want more control over what you're plotting, like if you want to plot a Circos plot, then you can move to R and use R functions and look into like Bioconductor has a lot of packages there that will help you visualize sequencing data. And I think I'm out of time, so I'm not going to go into our visualization example. But if there are any questions about alignments or the way I created those bash scripts, please ask me now in the chat. And other than that, I guess class is over and you guys will see Igor next week, I think.